Hi everyone, continuing our discussion from the previous time, we were discussing one of the ways you can do evolutionary experiments in lab with bacteria. We'll start our discussion with the second way you can do these experiments in a lab and then move our discussion forwards as to what is the kind of information that we can look at from these experiments and theoretically try to come up with analysis which might tell us something about these experiments and making us being able to predict what might happen in these populations. Okay. So, the second one, the second way you do these experiments is using something which is called a chemostat. So, chemostat is essentially just a stirred reactor. So, you have what this means here, that the contents of this reactor are well mixed, which means there are no spatial heterogeneities associated with concentrations. Concentration of every species, nutrient inside this reactor are constant everywhere, and you have bacteria growing in this environment. Now, the problem with the test tube experiment is that, as we had discussed last time, that you grow test tubes at a particular time, you add n naught number of individuals and you let growth happen for a day, and next morning you come to the lab and you transfer, and the number of individuals in the test tube has gone up to nt, and from these nt number of individuals you pick up n naught and transfer them to the next tube, and the process repeats itself every single day. The main problem associated with this type of experiment is that the condition in which growth happened for those 24 hours does not remain constant. In the beginning few hours of the experiment, the conditions of growth for bacteria were very good, they were not very high in number, the nutrients were plenty, and hence growth happened at a particular dynamics. However, as you move forward in time for that one day in which growth takes place in a test tube, the conditions associated with the experiment change quite dramatically. Nutrients become depleted, the waste from metabolism of the individuals gets secreted into the environment, you have cells entering stationary phase of growth, and as a consequence of all of these, you have physiology associated with the cells changing with time. That is not really ideal for an evolutionary experiment, because one of the main things that you want to test is that how do my bacteria adapt when I grow them in this completely defined environment E that I am making in this test tube. However, because of the points we just mentioned, that environment E does not remain constant in a test tube type of a setting. Hence, we move to something which is called a continuously stirred reactor or a chemostat, which alleviates that problem and helps you maintain constant environmental conditions at all times, which truly gives you a measure of how well bacteria adapt in this defined environment. So, this reactor is going to have a volume V associated with it, and it has a particular defined environment, and you have stirring taking place to make sure that there are no heterogeneities of concentration in space. But this is still a test tube. This still looks like a test tube experiment that we talked about, the only difference being that there is a stirrer and this is larger in volume. But what alleviates that problem is that you have an incoming stream to this reactor at the rate V naught volume per time, which is carrying fresh media. So, this is the media which you desire to do your experiment in, and you have a continuously you have this stream carrying these nutrients continuously at rate V naught. So, this would mean that the volume of this reactor increases at the rate V naught, but to alleviate that, you have an exit stream which is removing media and cells from this reactor at exactly the same rate V naught. What if you do this for a long enough time, what these conditions ensure is that the conditions inside this reactor. So when you do this experiment for a long time, analysis of a reactor such as this shows that the conditions in the reactor become constant. They do not change with time, and by conditions we mean the associated concentration of every nutrient, what is the number of cells in the environment, and so on and so forth. Every single one of those aspects associated with life in this reactor becomes a constant with time. And what's more, the conditions are equal to that in the exit stream. So this represents conditions in the environment, in the reactor. Because the analysis of this reactor would show that the exit stream is carrying what is present well mixed inside this reactor. So now you have this reactor like setting which gives you constant conditions with time because of these exit and entry streams. One of the consequences you should realize of this is that the fresh stream that is coming in, that is only carrying the incoming stream that is coming into the reactor, is only carrying fresh media. It's not carrying any bacteria. Whereas the exit stream, which is again going out from the reactor at rate V0, is carrying bacteria and spent media from inside the reactor. And what we are saying is that if we do this experiment for a long enough time, the reactor establishes a steady state associated with it. What that means is that the number of bacteria which are removed by the exit stream, which is flowing at volume V0 per time rate, the number of bacteria removed by this stream per time has to be exactly matched by the number of bacterial divisions taking place inside the reactor, such that the number of bacteria inside the reactor does not change with time. So that equilibrium also gets established between these two variables. So you also know the rate at which division is taking place inside the reactor at any given point in time. So again, you do this experiment for a long enough time, you maintain your incoming stream for a long enough time, and you keep taking periodic samples of what is present inside the reactor from that exit stream, and that gives you a snapshot into what is happening after 50 or 100 generations, or after however many generations you may choose to take your freezer stocks. So that's a second way that you would do your experiment. This is also called the analysis of this type of a reactor is also referred to as a continuous stirred tank reactor. Which is CSTR. Of course, doing an experiment like this would mean that you have to maintain sterile conditions for the reactor for however long that you plan on doing the experiments, but it offers you this great advantage that the environment associated with the experiment is now truly constant, which wasn't the case with the test tube setting that we discussed in the previous lecture. All right. So now if we do an evolutionary experiment via any of the two ways that we have discussed and try to answer a simple question, that in an evolutionary experiment, what do I expect will happen to this graph? On the x-axis, I am plotting time, and let's again represent that in number of generations. And y-axis, we represent fitness, mean fitness of the population. Which means if I have n different genotypes, the mean fitness is simply represented by sigma xi fi, where xi is the fraction of population of genotype I, and Fi is the fitness of genotype I. This is true if you are if the population is carrying n distinct genotypes in the environment. So, we start our experiment by a set of individuals which are isogenic and are all at fitness F naught. So, if you are starting with a genetically identical population where all individuals have this fitness F naught, then the mean fitness of the population at that time is also going to be equal to F naught. Starting from this point, I would like you to sort of pause this video for a minute or so, think about this graph and qualitatively sketch as to what you would expect as you go forward in time to happen to the fitness variable associated with the population. So, just maybe take a 2-minute pause, think about this and try to come up with intuitive solutions to this graph that you can think of. You can think of more than one ways, you can think of more than one logical ways or logical graphs that appear reasonable to you, and try and come up with reasons that you would expect to happen to this graph. All right. So, from a number of experiments, evolutionary experiments that people have done, it is found that the nature of this graph as we forward in time looks something like this. What's going to happen is that initially you have a lot of beneficial mutations which are available to you. So, the individuals keep picking those beneficial mutations and the mean fitness keeps increasing with time. Eventually, however, you run out of beneficial mutations to be conferred to the population, and then the fitness can no longer increase and it plateaus with time. Another way to think about this is in the context of fitness landscapes that we have discussed earlier in the course. So, remember during fitness, when we were defining fitness landscape, you have sequence spaces which are connected by edges and nodes. Nodes represent sequences, two sequences are connected if they differ only by a single nucleotide, and the third axis corresponds to the fitness associated with that particular genotype represented by a node. So, in a simplified one-dimensional sequence space, this is fitness. We had seen that we could get many types of fitness landscapes, but let us imagine that this is the fitness landscape that I am talking about. And now in the evolutionary experiment that I am starting with, this is the point where fitness of the n individual was F naught, and this is the genotype of my starting cells. Everybody in the starting population had this particular genotype and this particular fitness. Now, what is going to happen is that individuals are going to keep acquiring these beneficial mutations, and the mean fitness associated with the population keeps increases, and eventually, as we saw, because of finite mutation rate, there will be a quasi-species equilibrium between the mean fitness, between the peak fitness and the neighboring sequences which are very close to the peak fitness. So that quasi-species equilibrium will establish itself very close to the peak of the fitness landscape, and that's what is happening in this intuitive picture that we have drawn here. When the experiment is just starting, there are a lot of beneficial mutations which are available to the cell. So instead of going this way, the population could also move along this face of the curve, or it could also move along this face of the mountain, and so on and so forth. So these mutations, there are lots of mutations available which increase fitness, and the cell is continuously able to access those mutations, and the rate of the fitness is sort of linear with time as we move forward in generations. However, as we approach the peak, the number of mutations which further increase fitness of the population dry up, and that leads to this plateauing effect of infitness, and fitness does not increase with time as it was during the initial phase of the experiment. So, you have this saturating kind of behavior that is observed experimentally when you monitor fitness with time in a defined environment. But what we can see here is that in these representations, we can define a variable called speed of evolution. And let me just redraw that graph again. So, this is time in generations and this is mean fitness of the population. And we note that starting from F naught, the population increases and then saturates. And one of the ways we can define a quantity called speed of evolution is equal to rate at which fitness changes with time. If fitness is changing very rapidly with time, that means you have rapid evolution taking place because the population is moving towards higher fitnesses in a very fast way. However, if change in fitness with change in time is happening very slowly, that means evolution is not taking place at such a pace. So, this can be defined as delta F divided by delta T, where delta F is just F2 minus F1, fitness at time 2 minus fitness at time 1 divided by T2 minus T1. And what this tells me is that if I were to choose T1 and T2 and on this graph find out its corresponding F1 and F2, then this change F2 minus F1 divided by this change T2 minus T1, that is what gives me an estimate of speed of evolution associated with the population. And this can be seen that this quantity is just equal to slope of this curve. Another thing you should note here is that in this region of the curve although it becomes nonlinear and has this saturating tendency, in this region of the graph the curve can be estimated as almost a straight line. Okay. So, in this straight line region we say that evolution is happening linearly with time. As time increases by a certain rate, fitness is increasing by a rate which is proportional to the increase in time. And later on, as you move towards larger times, that rate slows down and eventually comes to 0 when this curve perfectly becomes horizontal. But we will be talking of this linear region where evolution is taking place at a linear rate or fitness is increasing at a linear rate with time. Okay. So, let us try and develop expressions associated with that. How do we even go about thinking something like that? So, in an experiment, if I am doing an evolutionary experiment, let us go back to one of our representations and see what does that tell us. So, we will go to the first representation that we did, where x axis represented time in generations and y axis represents number of cells from 0 to n. So, first thing that I want to imagine is that how would evolution happen in this chemostat type of a setting where I am doing my evolutionary experiment. If I were to just imagine this in my head, I know that a lot of mutations are going to get lost, and those are going to be represented in my graphical setting as follows. I have a beneficial mutation happen and it goes away by drift. This now represents in my chemostat as those mutations being washed away from the reactor to the outside before they had a chance to spread into the population. These are beneficial mutations which are happening, but were not able to spread because of drift. After some time, I have another beneficial mutation not surviving, another beneficial not surviving, and so on and so forth. Eventually, I will get one beneficial mutation which is able to get established. You should always remember the three fates of a population: extinction, establishment, and fixation. So, eventually you have a mutation that gets established, and once it is established, that means random chances are not going to be able to eliminate that mutation, and now selection takes over, and selection ensures that this mutation which has gotten established, which by definition is beneficial in nature, is able to go to fixation. So, you have this establishment taking place, and then you have mutation going to fixation. And then the fate repeats itself, that you keep having these beneficial mutations, but they do not survive, and eventually after a while you have another beneficial mutation which may survive and get established, and once established it goes to fixation. So, that is the intuitive picture that we have of evolution taking place of a microbial population in an experiment. Now, what we want from this type of an output is the slope of this curve, which gives me the rate of evolution or the speed of evolution associated with this microbial population. That is what we are going to work towards. So, the first thing that we want to understand is there are two quantities here, that what is the waiting time T1 such that a mutation gets established Remember, mutation getting established is referred to as the number of individuals which belong to that particular genotype exceeding 1 by S, where S is the selection coefficient associated with that particular mutation. So, what is the time that is associated with mutation getting established? We are interested in this because if a mutation is established such as this one, it means that it can no longer be eliminated because of drift, and now selection ensures that the numbers associated with its frequency go up to 1 and the parent genotype is eliminated. How long do I have to wait for this to happen? The second time that I am interested in is, it is called that T2, and that time is what is the time it takes for the mutation to go from establishment to fixation. How much time does a mutation need to go from this establishment event take place here and the fixation event take place here? This time is referred to as T2. So, these are the two times that I am interested in, which will tell me because I can see that fitness of the population has gone up by the magnitude of this beneficial mutation. Fitness of all the individuals has gone up. So, let me rephrase that. At the beginning of this experiment, at this time, the fitness of every individual in the population was 1. Suppose the mutation that happened here has a selection coefficient S0 associated with it. That means fitness of every individual which is carrying this mutation is equal to 1 plus S0. If that is the case, when the mutation gets fixed in the population at this point, fitness of every individual in the population now is 1 plus S0. Since the fitness of every individual has changed from 1 to 1 plus S0, that means the mean increase in the fitness of the population has gone up to 1 plus S0, and the mean has increased from 1 to 1 plus S0. That means a change of S0 has taken place in time T1 plus T2, and that gives me the speed of evolution in that sense. So, I need to understand that I need to get an estimate of T1 and T2, and I know that in the time T1 plus T2, S naught fitness changes by S naught. So, in that sense, speed of evolution is just given by S naught divided by T1 plus T2. But before I get that, I have to understand that how are mutations happening in the population. What is the probability associated with mutation of which confers a fitness of S naught, which confers a fitness advantage of S naught to an individual. Intuitively, what do we believe? Let's try and picture this graph. On the x axis, I have S, which is the selection coefficient. And this graph starts from a value of S equal to 0. I am not interested in negative values of x, because negative values of x mean that this is a deleterious mutation that has happened in the environment, in this mutant. And deleterious mutations are not going to get selected for, selection will make sure that they are eliminated. Hence, I am not going to worry about deleterious mutation, I am only interested in beneficial mutations. Beneficial mutations, which would mean S is positive. On the y axis, what I want to represent is that probability that a beneficial mutation confers an advantage of S. So, what we mean here is that this is a probability distribution curve, and what we mean here is that this is S1, this is S2, this is S3 and so on and so forth. This is increasing S here. Do we anticipate that every time a beneficial mutation happens, is its magnitude more likely to be S1 or S2 or S3, or is its magnitude likely to be equally distributed, or are beneficial mutations which confer a large advantage in fitness are more likely to those which confer a small advantage, and so on and so forth. The essential question is, what is the frequency distribution associated with beneficial mutations that we know of? And this is important because when we are interested in rate of evolution, we want to understand this graph, and this graph tells us that the speed of evolution can just be given by S naught divided by T1 plus T2. But we don't really understand what is S naught. More importantly, the probability that any mutation is able to survive drift and is going to establish itself, this probability is itself dependent on S, and in fact that probability is equal to S, as we saw a couple of lectures back. So, understanding the fitness advantage conferred by a beneficial mutation is extremely important for us to be able to comment on the nature of the dynamics associated with a particular mutation. Before we end this lecture, I want you to just imagine this scenario, that if we were to think of the magnitude of a beneficial mutation on x axis, such as shown on this graph, and on the y axis the probability distribution of a beneficial mutation of a particular magnitude happening, what do you imagine that graph would look like? And this is understanding this graph is important because of very obvious reasons that we just discussed. So, again, after end of this lecture, I want you to think about this graph and draw intuitive pictures that agrees with the understanding of an evolutionary process that you have in your mind, and we will start the next lecture starting with a discussion on this particular graph. Thank you.